So I will address the problem of microtubule targeting agent, and we will see a couple of issues about these chemotherapy drugs. So why microtubule targeting agents? It's because microtubules play a crucial role in many biological processes, in particular in cell division, but also in intracellular transport. And if you look at the right side on the video, you see that these microtubules are filaments that are highly dynamic. And these targeting agents will perturb these dynamics and induce perturbation in cell division when they are given high dose. So at high dose, microtubule targeting agents are known to be chemotherapeutic drugs. But in fact, what is interesting for us, and especially for Stefan, was to understand what was the action of this drug at low dose, because they observed that this drug had an anti-angiogenic effect. So if they address the drug to endothelial cells, they observed that there were a reduction of the migration, and they also look at other cells on epithelial, on glioblastoma cells, and they observe for these cells an anti-migratory effect on the cells. Small effect, but at least minus 20% less of migration. So the question of Stefan was how these drugs can have such an effect at low dose, and are we able to model this effect on the migration? So the mathematical issue will be describing the dynamical instability of the microtubule, and to address the problem of modeling the effect of the drug, and also look at the problem of cell migration. Okay, so I will start with the problem of description of the microtubule instability and the action of the MTAs, and then we will see the problem of describing the action on cell migration. So what are the microtubules? Microtubule is part of the cytoskeleton characterized by the stability. So on the left, you see in the cells the family of microtubule with lots of activity. And on the right, you see a cartoon where you see one filament of microtubule. These filaments of microtubule are long cylinder of diameter 25 nanometers. And these tubes are made of about 13 protofilaments. And each protofilament is just an assembly of alpha-beta tubulin dimers. So the assembly has a plus end and a minus end. The assembly is always alpha-beta, alpha-beta, alpha-beta. The end made with alpha is called the minus end. In the cells, this end is attached to the centrosome, so it has no dynamics. But if you look at the beta end, called the plus end, this end, plus end, has a highly dynamics. In fact, these dimers are in two different energy states. One state is more stable than the other one. The stable state is called GTP, whereas the unstable state is called the GDP. And if you look at the cartoon, when the tube is open, in fact, you can imagine that the open part of the tube is a kind of stabilizing cap. And when this stabilizing cap closed, you see that the microtubule depolymerized. What is known, in fact, is that this stabilizing cap is mainly made of the stable type of dimer called the GDP. So if you simplify the vision of a microtubule, the microtubule can be seen as a tube made first by the unstable state of the dimer, the GDP, in red in the slide, and a stable cap called the cap, the stabilizing cap of the microtubule. The main reaction that holds in this system is that we have polymerization. Polymerization is the addition of GTP, the stable one, on the cap of the microtubule. After some time, the GTP in the microtubule are hydrolyzed. Hydrolyzation is transformation of GTP in GDP. And so we have the cap. The evolution of the cap is due to two reactions, a reaction of polymerization and a reaction of hydrolysis. When the hydrolysis rate is bigger than the polymerization rate, then the cap starts to diminish. And when it disappears, then the microtubule is able to depolymerize. Depolymerization gives rise to new GDP and stable dimer of tubulin that I will call free GDP. And after some time, these free GDP are recycled in free GTP that will be able to get polymerized. So we have a cycle, and we were able to modelize this cycle. So what we have in hands to calibrate and to study this system is the follow-up in cells of the microtubule. In fact, the biologists are able to label the GTP cap thanks to end-binding proteins and green fluorescence protein. Then, in fact, in white here, you see essentially the GTP cap of the microtubule. And thanks to software like Tip Tracker, we are able to follow the evolution of each microtubule and to have information on the main indicator of the dynamics. In fact, indicator of the dynamics are growth lifetimes, growth distance, growth speed. With this information, we can build information what are called catastrophe frequency, either in temporal or spatial form. But we have also information less accurate, but we have information about what we call shrinking lifetime, shrinking distance, shrinking rate, and so have information on what is called rescue frequency, temporal or spatial frequency. So with this, we will be able to try to calibrate a model if the model is able to represent this macroscopical indicator of the unstable instability dynamics. So in fact, here I also show you what is called a chemograph. So chemograph is, if you look in the vertical, you have the time. And if you look at each time of the image, each line of the image corresponds to the evolution of a microtubule. So you see in white the cap, the microtubule cap. And so you see that first the cap increase. In fact, the distance to the boundary increase. So you see that the speed rate, polymerization speed rate is almost constant at the beginning. It's about the same about the hydrolysis. And after some time, you see that the cap disappears. The disparition of the cap corresponds to a catastrophe. And if you look more carefully to this chemograph, or to look carefully to many chemographs, you see that the disparition of the cap can be due either to a slowdown of the polymerization, that can be modeled by the fact that you don't have any more, or not sufficiently, not a sufficient amount of GTP tubulin to be polymerized, or you also see acceleration of the hydrolysis speed. And the novelty that we introduced in the PhD of Ayuna Barlukova was to take into account this, what we call aging of the microtubule. So we started from a work from Ino and his collaborator that was proposed in 2009. The model were constructed thanks to structure population models. So this is a coupling between two structure equation and two ODEs. So the population of tubulin were split in four: the microtubule in polymerization, the microtubule in depolymerization, and the free GTP and free GDP. Free GTP and free GDP can only be structured by the time. And if we look at the population of microtubule in polymerization, there are two more traits that are important: the size of the microtubule, but also the size of the cap. For microtubule in depolymerization, we don't have any more the cap. So the population can be structured by the time and the size of the microtubule. So it was the proposition of Ino and all. And what we propose with Ayuna to better model the aging, the phenomena of aging, but also to better model the growth indicator, the idea was to add an additional trait, that is the age of the microtubule. What is the age of the microtubule? For a microtubule in polymerization, the age is the time after the last rescue. And for the microtubule in depolymerization, this is the time after the last catastrophe. So thanks to that, we adapt the model of Ino and Hall, and we can have a look to the type of equation. So here is the equation for the population of microtubule in polymerization. So we have the time, the size of the cap, and the age. The velocity of the age with respect to time is equal to 1. The velocity of the microtubule size is due to the polymerization, and the polymerization speed is linked to the number of GTP available in the system. So we take a function gamma pole that is 0 at the beginning, because if you don't have enough GTP, you cannot polymerize, and it saturates. It's linear than saturation. Then, if you look at the last coefficient in the equation, you see this is the velocity of the cap. That is a balance between the polymerization speed and the hydrolysis speed. And the hydrolysis speed depends on the age of the microtubule. Then you have associated to this equation boundary layers, boundary condition. One boundary condition corresponds to the nucleation process, that is addition of new microtubule. The other corresponds to rescue event, And we have an age boundary condition to close the system. So here are the 3 other equations. You see that the polymerization is modeled thanks to a transport term 2. So we say that the microtubule is able to depolymerize at a constant speed. And we have in the left-hand side of the equation for V, the apparition of 2 terms corresponding to a rescue event and catastrophe event. And finally, in the last 2 equations, the equation for P and Q, in equation of P, you find a decrease of the population due to the polymerization, increase due to recycling, and again, decrease due to the nucleation term. And in the equation of Q, you see the red term is the augmentation of the population due to the depolymerization and loss due to the recycling. At the end, if you look at the total amount of tubulin, you will see that the total amount of tubulin in the media will be conserved. Okay. So thanks to that, IUNA was able to calibrate the system and to mimic a dose effect. So here is a dose effect of Taxol. Taxol is a polymerization drug. And what she showed and what she made is to build a strategy to calibrate all the parameters of the system. You see that we have a big system with many parameters, so it was really tricky to calibrate it, but she succeeded. And on the right, you see the data, the macroscopic indicator of the instability in this graph. So here we have 4 indicators of the stability that we used: the growth rate, the depolymerization rate, gamma dipole, and the frequency, temporal frequency of rescue and catastrophe. And we put this indicator on the 4 alpha axes in the plane. So with no drug, we put this indicator to plus minus 1, and we model the effect of the drug on each of these indicators. And what you see is that this evolution is non-monotone with respect to the dose of drugs. And here, what you see is the effect that even for very low dose, 1 nanomole of drug, you see a big effect of the drug on the frequency of rescue, for example, but not only, also an effect on the frequency of catastrophe. And so what proved IUNA is that this small effect, this effect due to the small dose, can be explained by an effect on 1 parameter that was RS in our model. And RS is just a delay linked to the time needed for a microtubule in depolymerization to go back to the polymerization state. And a small effect on this parameter can explain large effect on the macroscopic indicator. So we were happy to have a small possible interpretation of this effect at low dose. Okay? So here is the work we've done with IUNA, but in fact we go further with Magali Tournus and Diana White, because we were not satisfied by the modelization of the depolymerization. In fact, depolymerization the depolymerization speed is bigger, much bigger than the polymerization speed. And we can see the depolymerization process as a sudden process. And so we decided to model it thanks to fragmentation model. So we changed the equation on V here, and the depolymerization is now modeled thanks to fragmentation term, and we are still working on it, and we simplify this model to try to look at the asymptotics of this kind of things. So when we will look at the cell migration process, we will have to go further because the localization of the microtubule will take a crucial role. And so you will see later in the talk how to build a very, very simple model of microtubule, but using the space dependency of the microtubule. Okay. So now cell migration process. So we will first look at the actors of cell migration, and I will focus on one model built by the team of Edelstein-Quéchet, and you will see how with Rémi Tesson we adapt this model to take into account the microtubule impact on the process. So first here you see two movies. On the left you have a control cell, so no drugs. And on the right you see cells with drug. This is a depolymerization agent here called vaflunin, if I'm not wrong. So you see that in that case, the migration speed decrease. You see that you have less contraction at the rear of the cells and less dynamical activity at the front of the cells. So you see the impact at low dose of one microtubule targeting agent on this type of cells. So what are the main engines of the motility? There are four. First, actin polymerization or nucleation at the front of the cells. Second, membrane tension of the cells. Then cells adhesion to the substrate. And then cell contraction. What I omit to say is that I will concentrate on migration on a 2D substrate. 3D migration is has a different type of process. So one of the main actors is the filament of actin. Filament of actin are other part of the cytoskeleton. And we see in the cartoon from the paper of Blanchois that the organization of actin can be different with respect to the position inside the cells. At the front of the cells, you see a branch cross-link network located at what we call the lamellipodium of the cells. You see also fingering of the cells that correspond to the organization of the filament as parallel bundles. And if you look at the back of the cells, you can see stress fiber that is parallel fiber, but in the other direction. The filament of actin are also polymerized with the plus N and minus N. The plus N is called the Barber N, and in the case of stress fiber, the plus N are on the other side. Okay. So these actin filaments are regulated by many, many proteins. And among these proteins, we can mention RP23, linked to nucleation and forming at the location of the filipodium. There are many proteins linked to sequestration, thymosin, frophilin, twyphilin, capping. In fact, this protein limit the polymerization of the actin, maintaining a pool of actin available, always available. There are also proteins linked to fragmentation and depolymerization. We can think about cofilin, gelsofilin, and protein linked to contractility that are mucin protein. And in fact, all these proteins are regulated themselves by Rho-GTPase protein. RAG protein at the front of the cells, Rho protein at the back, at the rear of the cells, and CDC42 for the filipodium part of the cells. So, to be short, we can say that RAG protein will promote the protrusion of the cells, so promote a force that enables the cells to go further. And at the back of the cells, the protein Rho will promote the contraction of the cells. Okay? So, the work of the group of Edelstein-Keschet proposed a very simplified view of this complex activity. And what she proposed is first to say that the cells is something can be modeled thanks to fluid dynamics. And this fluid dynamics, if you look at the Reynolds number associated to the cells, you can see it's very low. And so the inertial force exerted to the cells are negligible with respect to the viscous forces. And so we can look at the system as the Stokes, thanks to the Stokes equation, with U that will be the velocity in each point of the domain, corresponding to the cell, but also outside the cells. And in the work of the group of Edelstein-Keschet, they say that there were two types of forces that exert on the cells. There were protrusive forces and there were elastic forces. They neglect the contractile forces in that vision of this simplified vision. So coupled to this mechanical model, we have inside the cell a biochemical model. And in the paper of the group of Edelstein-Keschet, they only consider one type of protein. You can think at the protein RAC. And this protein can be either in an active form or in an inactive form. So this protein will evolve inside the cell thanks to a transport term due to the fact that everything moves at the velocity u, diffusion process, and reaction terms that explain how the protein will be activated or inactivated. Okay? So starting from this work, we decided to improve a little bit the description of the biochemical model. So first of all, we will add the Rho protein in the system because we want to take into account the contraction forces at the back of the cells. So if you look at the Stokes equation, we have an additional term corresponding to the contractile forces. And then we also add a model that will represent the action, the presence on the microtubule. So we are not able to couple all the precision we had in the model of Ayuna here, and we decided to limit us by having a quantity that will model the amount of tubulin in each point of the cells. And we suppose that we had a certain number of microtubules, n microtubules, somewhere at the front, somewhere at the back to handle the contractility. And for this microtubule, we keep the position of the cap and the length of the microtubule. Okay? And we decided not to keep the approach of the group of Edelstein-Quéchet. Edelstein-Quéchet represent the cell, the membrane, thanks to marker cells. And we prefer to use a level set approach. So level set approach consists to say there is a function phi, and the membrane of the cell is the zero level of this function phi. Inside the cell, phi is negative. Outside the cell, phi is positive. So let's come back to the chemical model. So for the protein, we find again the same type of equation that we have in Edelstein-Quéchet. I will be more precise on the reaction term GP here. So for example, if you look at the reaction term for the RAC equation, you see that in red we have a localization term that say that we will have activation of the protein RAC only in the neighborhood of the cap of the microtubule. In blue, you have the classical reaction term that you can find in Edelstein-Quéchet, with a slight difference that it will depend on the presence of microtubule and tubulin. And if you look in green, in the desactivation term for RAC, you see that we put a competition term that say that in presence of Rho, we have desactivation of RAC. It was one of the main property that Stéphane Honoré tell us about the protein Rho and RAC. So a term that is quite near from the one of Edelstein-Quéchet, but with localization and competition between Rho and RAC. Now, if we look at the tubulin concentration, the tubulin concentration will follow a transport reaction diffusion equation. Transport to the speed u, diffusion, and with the reaction term that say that we lose tubulin due to the polymerization, and we have polymerization in the neighborhood of the cap of the microtubule. So we have this delta function here. And you see that the velocity of polymerization is something that is taken here linear with a concentration, threshold concentration Cc that will play a crucial role in the effect of the drug in a moment. In fact, here you see that the depolymerization of the tubulin is linked to the parameter Cc. So we have a linear velocity, and if the tubulin is less than this concentration, the microtubule depolymerized, and if not, it polymerized. And this concentration is linked to what we call the equilibrium linked to the dynamical instability. We have dynamical instability, but we have dynamical instability around some equilibrium. Okay, and here Cc is this equilibrium. Okay, so now the position of the microtubule. We have two types of microtubule that are on the front of the cells, microtubule that are in the back of the cells, plus for the front, minus for the back. And we have a region in neighborhood of the membrane where the microtubule are more unstable than far from the membrane. So in fact, we split the population in four type of microtubule. So the evolution of the microtubule, the position of microtubule will depend on the velocity of the cell U, but will depend also on the gradient of the tubulin, the gradient of the protein rack at the front, and we'll have a different orientation if we are in the front or in the back. And finally, the length of the microtubule is just linked to the velocity of gamma pole and dependent on the position of the microtubule. So, with this, we can come back to the forces that we had in the Stokes equation. We had three forces. One force that is elastic forces. The elastic forces took the form that we can find in the work on Côté Maître in a paper of 2008. So it depends on the level set function phi and is located on the neighborhood of the cells. Protusive forces and contractile forces are also located in the neighborhood of the membrane, and they're both oriented with the normal of the membrane. The normal of the membrane is gradient phi over the norm of gradient phi. And the intensity of the force depends on the presence of the protein rack at the front and rho at the back, with different type of shape that can be found already in the paper of Edelstein and all. So to calibrate this model, we built with the group of Stéphane Honoré a couple of experiments, and especially we had an experiment where they get either on the same cells the microtubule dynamics indicator and the migration indicator also on the same data. So these cells were either control cells, either treated thanks to vincristine. Vincristine is a depolymerization drug too, like the vinfluminine where we saw the effect in the first slides. And so we had information on microtubule dynamics after the information of cell migration, but also before them. So this is information for the dynamics of microtubule and cell migration, but we also add information about the protein rho and protein rack. They made what they call collimetric ELISA assay, and they were able to quantify the amount of protein rho and the percentage of activation of rho and rack. Unfortunately, we didn't have the amount of protein rack with such experiments. So, with this, we can look at the effect of the drug. You see that if you concentrate in the red table, after 3 hours, we have a deep effect on the migration speed of the cells. You see that if you look at the red table, you see that the growth speed of the cells decrease, and you see that the catastrophe frequency increase after the effect. So we try to reproduce this phenomenon, and here are the results, the output of the ELISA cells. So you see that the drug decreases the total amount of protein rho, and you see that the drug decreases the activation of the protein rack, the rate of activation of protein rack. Okay. So here is a simulation of Remy starting from an academic cells that is circle cells with a certain amount of microtubule at the back and at the front. And you see that the shape taken by the cells is quite reasonable. You can see here, after 3 minutes, the evolution on the left of the rack concentration and on the right on the rho concentration. And we can also model the effect of these drugs, so these increasing. So what we've done is to say that the drug will increase the stabilizing zone of the drug, so the parameter which is called distab was increased, and the drug will perturb the equilibrium, and in fact will increase the concentration CC. So we increase we decrease the speed, so we increase the CC, and we can look at the resulting data. So to conclude, I show you in 2 types of example how to take into account a low dose effect of MTAs. So with a quite complex model to take deeply into account many aspects of the instability process. And for the motility of the cells, we are more rough. It's more academic model, but it was a first step for us to learn about cell migration at that time. Thank you very much.
Thank you, Florence, for the clear and interesting talk. Are there any questions? Yes, Luis.
Thank you for the nice talk, Florence. I had 2 questions. One is, in the chemograph you showed, what were you marking with fluorescence? Yeah, this, because you seem to have the signal fading out before you depolymerize. So, you seem to have that the signal that you're marking is fading out before you start depolymerization.
So when you look when you look the cap, you have a depolymerization, and so you don't see the microtubule.
Okay.
And then you find again the microtubule when we have a rescue, when the microtubule polymerize again.
Okay, so what you're marking...
The fluorescence is only able to mark the GTP.
Okay, that's it.
The GTP.
And so you see it fading out before
so when it disappears, you have a catastrophe.
But it doesn't seem to disappear at once. It seems to fade out, so it doesn't seem to be that discontinuous.
Yes, you have some default on the cap before. It depends on the chemograph.
Okay, it's not a typical thing. So here I choose
a chemograph where you had this default to see that we may hypothesis that aging of the microtubule
makes it fade out.
Yes, okay. But this is something that was observed many times by Stefan. So
Okay, it's linked to the second question, which is, I'm a bit surprised that you can model this as a fragmentation process, because when you break into two fragments, you have that your microtubules, one of the fragments is linked to the centrosome, but the other one is free.
So in fact, I talk about fragmentation, but my microtubule fragmentation consists in a shorter microtubule with many GDP, free GDP.
Okay, so
So
the second fragment just disintegrates right away.
Yes, the second fragment disintegrates.
Okay, okay. No, then it makes sense. Thank you.
Are there? Yeah.
In your last part, do the microtubules interact with the cell membrane? Are they pushing on it or are they pulling on it in your case?
Sorry?
Do the microtubules interact with the cell membrane if they go all the way? No, they don't interact at all. No. They neither push nor pull on the cell membrane.
No, I don't think so. I have no...
Any more questions?
No, the force induced by the protein is linked to the fact that the protein will activate the polymerization of actin, and so it's a rough description.
If it interacts with the membrane, it could change the catastrophe.
Yes, yes.
Any more questions?
Where do you get the nice properties of the level sets of your function phi so that it doesn't split into different components? It comes from where?
We stopped before. The computation is very long, so we don't see
your level set splitting.
No.
But there's nothing in the equation to prevent that.
Probably, but the computation was so long that
okay.
we did not reach this time.
Okay, thank you.
I do have a quick question. In the second part of the talk, you assume the functions gamma and g to be linear.
Gamma
and g at some point there were. Next slide, probably a couple of slides after.
A gamma point.
Yeah. How much do you think that affects the results? Would the results change a lot if you change the definitions for those functions, or?
If the concentration of the tubulin stay around the concentration CC, it won't change a lot.
Much. Okay.
Won't change a lot.
Okay.
I'm not sure in the model of Ayuna we see really the plateau.
Yes, yeah, I understand that. Okay, makes sense.
Yes.
Okay, thanks. Any questions on that?
So there is a question online. So the question is, you use the term catastrophe. Is it connected to some catastrophe theory models?
Catastrophe?
Catastrophe.
Alors, catastrophe is catastrophe in English, so sorry for that.
No, but is it connected to the catastrophe theory?
No, I think it's the vocabulary that Stefan used. He always, when the microtubule depolymerizes, he say it's a catastrophe.
It's not connected to the theory from the 70s or whatever.
No. It's just the vocabulary he used.
Thank you.
Thank you. We are a bit late. Shall we resume in 10 minutes instead of 15? It's okay. Yeah, we will resume in 10 minutes then.